community organizing it at IMALS. And we're very excited to have with us tonight two incredible doctors who are going to go over current ALS clinical trials and sort of give us an overview. And hopefully we'll have some, like I said, have some time for questions at the end. So we're going to ask folks to see if they can keep their questions to the end. And if we don't have time to answer them, we'll do our best to get an answer back to you after this webinar. We're also going to be recording the webinar for people who can't join in person. So just want to let you all know that we will be recording and we'll be able to put this on our YouTube channel to share with others as well. And so we just, again, want to thank you so much for coming. We had a lot of interest in this, so we're really excited to talk about the current ALS trials and also just get a sense of what's happening in the landscape. So first, also just want to say for IMALS, for those of you who aren't familiar with IMALS, we are a patient-led community and sort of have a threefold mission. The first part is to provide critical support and resources to those living with ALS, their caregivers and loved ones. The second is to really revolutionize how we cure ALS by empowering and mobilizing advocates to raise mainstream awareness. And the third is to engage with policymakers to drive the development of cures. So that's really what we're focused and on at IMALS. And then for the clinical trials team, which is one of our community teams that is led by our community leaders, one being Diane, who I'm going to introduce in a minute. The clinical trials team partners with researchers, government agencies, and drug sponsors to ensure access, efficiency, and humaneness of ALS therapy development and approval. So that's a main focus of our ALS clinical trials team. They're the ones who put this webinar together and have done a really great job of creating some community focused clinical trials information. And they also collect and distribute clinical trials information to the ALS community. So just wanted to give you some background in case some of you are new to IMALS, and we're really happy you're here. And just a few other housekeeping pieces. So you can use the chat feature, but like I said, we're going to try and keep questions to the end. We're also asking everyone to mute themselves while we're presenting to just make sure everyone can hear. And if you have any questions, like I said, we don't get to them, we're recording the questions in the chat and we'll be able to send you some answers after this webinar. But now I would like to turn it over to one of the wonderful co-chairs of the clinical trials team, Diane.
Thanks, Julie, and welcome everyone. Thank you, Dr. Bedlack and Dr. Li, for taking the time to speak with everyone tonight. Really looking forward to it. And one of our team members, Jill Bratton, played a role in setting this up, and we appreciate that. And one of my two fellow co-chairs, Dan McIntyre, is also on this call. Bob Hebron, our third co-chair, became a grandfather of twins yesterday or today. So Bob is missing that call. And so wanted to just take a moment to give a quick bio of Dr. Bedlack and Dr. Li. Dr. Bedlack is the Stewart Hughes and Wint Distinguished Professor of ALS. He's the director of the Duke ALS program. He's a leader of ALS Untangled, ALS CRLI, and the ALS Reversals programs. He has been working for 21 years to create new options and more hope for people with ALS. Dr. Li is an assistant professor of neurology at Duke with interests in neuroinflammation and antisense therapies. She's the lead author on a recent review of ALS trials, and I'm going to drop the link to that article in the chat so people can take a look at that. And I'm going to turn it over to Dr. Bedlack and Dr. Li. And again, thank you again for your willingness to be here today. We appreciate it.
Well, it's our pleasure, Diane. Hello, everyone. Thanks for having us tonight. And it's a pleasure to be invited to speak to you about current ALS clinical trials, especially since I get to work with Xiaoyan Li. So you heard Xiaoyan's bio, but let me just tell you, she's the most amazing person that I've worked with in all my 21 years in ALS. I mean, nobody works harder than her. I thought I worked hard. You can see I'm in my home and she's still in the office. And she's also a lot smarter than me, as you'll see. So Xiaoyan, thank you so much for taking time to join us tonight.
Sure, thank you.
Yeah, so this is how I thought we'd organize tonight's talk. I'll make a few opening remarks about clinical trials. Xiaoyan will take you through a table that lists the clinical trials that are enrolling in the United States. Then we'll each take a turn describing the ones that we're most excited about. I'll tell you about some things that I see coming down the road soon that I'm excited about. I'll make a few closing remarks, and then we'll take some time for your questions. So just a few opening remarks. We're talking about clinical trials tonight. I mean, this is the clinical trials subcommittee of IMALS. I'm assuming most of you know what a clinical trial is, but just in case, a clinical trial is a research study, and it's one in which one or more human subjects are prospectively assigned to get an intervention. That intervention could include a placebo, could include a drug or a device, and the purpose is to evaluate the effects of the intervention on health-related biomedical or behavioral outcomes. So that's the official NIH definition. Trials are not easy, especially in ALS. There's a lot of challenges. I mean, to start with, as you know, this is a rare disease. And although we're doing better, funding and the numbers of eligible participants are definitely less than we see in other more common conditions like hypertension, diabetes, cancer. To make matters worse, many people who live with ALS are far from the sites that offer trials, and travel is not easy. We don't understand what's causing most people's ALS, and so we have uncertainty about exactly what to target. And in fact, a lot of us, I know Xiaoyan and I feel this way, don't think ALS is one disease. We think there are subsets of ALS that have different causes, that there might be different things driving progression in different patients, and we don't really know yet how to categorize most of these. We also don't have great ways to measure responses to ALS treatments. As many of you know, the most common thing we use is a clinical measurement called the ALS Functional Rating Scale. And it has been used to get a few drugs approved, but certainly I think most of us realize that it's noisy and it's very slow to change. I also noticed that there can be conflict between what some of us perceive to be the most scientific trial designs and what some perceive to be the most humane designs. For example, the question about placebos often comes up in discussions of trials. ALS trials take an awfully long time, and I've seen trials that take 2 to 3 years between when we get an idea, when we get it open, when we complete, and when we finally can disclose results. And as you know, that's the lifespan for many patients with ALS. We have competition for patients in our trials. It comes from what I call alternative and off-label treatments. I abbreviate those as AOTs, things being advertised on the internet to slow, stop, or reverse ALS, usually without any science. And that's a big problem because there aren't any internet police to hold people accountable for what they say about these kinds of products. And then this last one is a pet peeve of mine, which I'll give you a potential solution for at the end. I feel like many people all over the world are not getting reliable information about their research and experimental options with this disease. It's important to note that in spite of these challenges, we are making progress. So I feel like over the 21 years that I've been in this field, there's so much more federal and private funding for ALS research just in the last 5 years than in all the 15 years before that. And that's really mainly because of you, the ALS advocates, that are on this call and all over the world that are working hard to make this happen. And as a result of all the funding, there's more trials. I think there's been more ALS trials that have opened in the last 5 years than in all the 15 years that I worked in this field before. And probably my favorite thing that I've seen in all the years that I've been in this field is that people living with ALS are finally partners in trials. That was never happening when I first started. And now I would say most trials have patient advisory boards working with them all the way through. And I would say that there's one example that shows what we can accomplish when we understand the cause of ALS. So we do understand one cause of one form of ALS. That's the mutant SOD1 form of ALS. We really have a good understanding of how that mutation causes disease. And we have a biomarker that seems to work quite well in that type of ALS, which is called neurofilament light chain. And because of those two things, we've had what I would call a smashing success, and that's the drug tofersen. So this was the first drug to ever receive FDA accelerated approval for use in this disease based on a biomarker. So if you remember the trial, it showed a significant reduction in the mutant SOD1 protein, and it showed a significant reduction in the amount of this neurofilament light chain. But really at the end of the trial, it didn't show an impressive effect on function. Nonetheless, the FDA was so convinced by those biomarkers that they approved it. And now, as we see what's happening to people who've been on tofersen for a long time, we're seeing that it's a home run. So about 50% of people who are on that drug for more than 2 years have stabilized. 20% are recovering lost motor function. So if someone stands up at a meeting and says it's not going to be possible to reverse ALS, you can point to this and say that it's happening. We just have to understand the disease better. So I'll turn it over to Dr. Li now to give you an overview of what's happening with clinical trials.
Thank you, Dr. Bedlack. So now I'm going to review the pathophysiology of ALS, and hopefully this will help us understand the clinical trial investigational products, which I'm going to show you in on the next slide. So so you can see that this is a very busy slide and it correlates with a very complicated pathophysiology of ALS. So far, aside from those genetic forms of ALS, we still don't have a very clear understanding of the disease causes of ALS, as Dr. Bedlack just mentioned. However, over the past few decades, we have gained a tremendous amount of knowledge in understanding ALS pathophysiology, mostly from studying this ALS causal genes, such as SOD1, C9 open reading frame 72, FOS1, and taxin2, and TDP43, etc. There are many of them, probably approaching 40 different genes now. And some of those genes regulate oxidative stress, such as SOD1, and some regulate neuroinflammation, and this include C9 or F72 and TBK1 and optineurin. And some genes are important to maintain mitochondrial dysfunction. And you probably know TDP43 and FOS and C9. Those proteins are very important in regulating nuclear cytoplasmic transport. And in addition, many genes are very important in many gene mutations are involved in the disruption of RNA metabolism and DNA repair, and also protein homeostasis. Protein homeostasis basically means that the process of maintaining protein level, protein structure, and protein function in normal cell. And we have seen several gene mutations disrupt this process. In addition, there are several ALS causal genes are very important in maintaining motor neuron axonal integrity, and also very important for axonal transport. So you can see disruption of any of those process can lead to ALS. And also the microenvironment of those motor neurons are also very important. And we know that excitotoxicity and oxidative stress, really those are the earliest hypotheses for ALS pathogenesis. And this, the study of the oxidative stress and excitotoxicity led to the FDA approval of the first 2 ALS disease-modifying treatment, including riluzole and radicava. So in this talk, we'll be discussing the investigational drugs in phase 2 and phase 3 trials that target this, various different mechanisms. Before the discussion, I also want to mention the gene targeting therapy because this is really the most promising ALS treatment because this really treats the root cause of the disease. Can I control the slides or you have to control the slide?
I have to control them, I think.
Okay, next.
Just tell me when to turn. Yeah.
Yeah. So, recently published article, you can find the list of emerging drugs in phase 2 and phase 3 trials for ALS. So in this slide, I want to draw your attention to the mechanism of action column. As I mentioned, you can see that various drugs target different disease mechanisms. Some of the product, for example, rapamycin, can, impact multiple different disease mechanisms, including protein homeostasis, autophagy, and neuroinflammation process, and many other medications also target different disease mechanisms. Since the publication of our article, 2 clinical trials have completed. This include B2, B105, and Tadalafil. Unfortunately, those 2 clinical trials were unsuccessful. So B105 is the antisense oligonucleotide targeting the ataxin-2 gene. So although in this clinical trial, the products shows it could affect the ataxin-2 protein level. However, neurofilament light chain level did not change, and the disease progression also did not show any difference between the 2 groups. So Biogen has since discontinued development of this drug. The other one is a phase 3 trial of Tadalafil. The study also did not show any benefit in slowing down disease progression. So in today's talk, I'm going to focus on 2 products, RAPA501, which is another name is called regulatory T cells or Treg. Another product is AIT-101. At the end, I'm also going to discuss with you about a new trial called ATLAS trial. Next slide, please. So a study published in 2018 on JAMA Neurology and found that the regulatory T cells or Treg cells inversely correlated with ALS progression. And there are multiple other studies have shown that in ALS patient, that their Treg cells, either their Treg cells numbers were reduced, or even if the Treg cell numbers were normal, their function were impaired. And based on those preclinical studies or some of the in vitro studies, or observational studies, there was a very small phase 1 study were designed to study the Treg cells in 3 ALS patients. So what they did was that they took the whole blood from those 3 patients and then isolated regulatory T cells, and then expanded those cells ex vivo. Ex vivo means that outside the patient's cell body. After the expansion, they gave those cells back to the same patient, and this process is called autologous infusion. And during this small phase 1 study, and they found that infusion of Treg cells were generally safe and well tolerated. And they also found that Treg cell numbers and their function level have improved. And in this small study, they have shown that increased Treg cells correlated with a slowing of ALS progression. But you can see that, well, this study definitely is limited by the very small N number. So since then, another phase 1, phase 2 study is completed. So basically, in this study, they tested multiple different dosing regimen of RAPA501, which is Treg. And they showed that this different dosing regimen were all safe for in the ALS patients. And currently there's an open label phase 2, phase 3 study still ongoing, and is actively recruiting patients. The goal of this study is, one of the main, goal of the study is to test the highest dosing of Treg cells to see whether it's safe in a bigger population. So another important outcome is that they're going to examine inflammatory biomarkers. It's mostly a biomarker study and a safety study, but certainly they will gather efficacy data as well. Next slide, please. So now we're going to switch gear. We're going to discuss about another product called Apilimod dimesylate. Another name is called AIT-101, or there's another name sometimes you may come across called LAMA, L-A-M-A, I believe. So there are a lot of research on ALS pathology. In the past few decades, we understand that in the majority of ALS patients, in their brains and their spinal cords, we see this TDP-43 protein inclusion or protein aggregation. This is demonstrated here in the cytoplasm. And this process leads to a loss of important TDP-43 function in the nucleus of the motor neurons. And also it leads to a gain of function, or it's a toxic gain of function in the cytoplasm. It's basically disrupt the motor neuron function, and ultimately to lead to cell death. In a few percentage of ALS patients, they don't have this TDP-43 inclusion, but they have SOD1 inclusion or FUS or FUS protein inclusions, and those are in SOD1 patient or FUS1 gene related ALS. Next slide, please. So you can see that because the majority of ALS patients have this TDP-43 pathology, TDP-43 protein aggregation or SOD1 FUS aggregation. If there's a way to sort of get rid of those protein aggregates, maybe that will benefit many ALS patients, not just few patients with certain gene mutation. So Apilimod dimesylate is one of those potential products that may help us achieve this goal. So AIT-101 is an oral PIK5 inhibitor, and in cultured motor neurons. So this product has been found to basically expel the TDP-43 aggregates or dipeptide repeat protein aggregates from motor neurons. And also in cultured motor neurons, epilimumab dimesylate has been shown to improve the survival of those motor neurons derived from ALS patients. Next slide. And based on all those previous preclinical studies, and a phase 2A study was designed. It was a randomized, double-blind, placebo-controlled trial in 15 ALS patients, and all of these ALS patients all have a C9 or F72 gene mutation. Those patients received 125 milligram either epilimumab dimesylate twice a day or placebo twice a day. There's 2-to-1 ratio for a short period of time, is 12 weeks, and followed by 12 weeks of open label extension phase. So this study was recently published. I think it was only a few months ago this year. So epilimumab dimesylate was safe and well tolerated in this 10 patients. The study also demonstrated orally administered drug was able to cross the blood-brain barrier and access the central nervous system. So the drug was able to inhibit PIK5 kinase activity, and was also successfully lowering the misfolded protein, which is the dipeptide repeat protein in this specific trial, actually by 73%, which is very significant. And certainly this result need to be repeated in a larger trial, the efficacy of this product need to be verified and established in further trials. Now, next slide, please. So lastly, I want to mention the ATLAS trial, and this is a very unique trial, a very novel trial for ALS patient. Although this trial is, is targeting SOD1 ALS patient. So previously, there are a lot of natural history studies in familial ALS patients suggest that there's a pre-symptomatic phase of ALS. So this is defined by the interval between the onset of underlying biological disease processes. Well, we're not able to see when we see the patient, right, or anybody. So between this biological phase and the appearance of the first clinical symptoms, so the interval between those two time points is called pre-symptomatic phase of ALS. And this phase may last from months to years, and you can see that it's very hard to identify this pre-symptomatic phase in sporadic ALS population. However, SOD1 gene carriers is a very valuable research asset in this study. So in this study, they enroll certain SOD1 gene mutation carriers, not every type, because there are currently about 200 different different type of mutations in SOD1 gene. They only enroll the mutations that have has proven to cause faster disease progression. So the participant will first enter into this part A phase. This is called a natural history run-in phase. So basically, this participant with a certain known SOD1 gene mutation will be monitored monthly. Their neurofilament light chain levels will get monitored. So at any time point, if their neurofilament light chain level increased above 44 picogram per ml, or there's a change of greater than 10 picogram per ml, and the participant is still is asymptomatic, and this participant will have an opportunity to get enrolled in this placebo-controlled phase. They plan to enroll about 28 patients, 28 participants, I should say. So those participants will get randomized to either tofersen group or placebo-controlled group. And during the Part B phase, if any of the participants start to show some ALS symptoms or confirm with ALS diagnosis, they will have an opportunity to enter into the next phase, Part C, which is open label phase. They will get tofersen treatment. And in case in Part A, if any participant develop ALS, they will directly enter into Part D, which is also an open label phase. They will get tofersen treatment. So, so Part B, C, and D will last up to 2 years in duration. So this study will help answer this question: does early initiation of tofersen delay emergence of clinical ALS or reduce the loss of motor function compared to later start of tofersen? So this is, I think this is a very exciting trial. It's very, very new. I will hand it over back to Dr. Bedlack.
Thank you, Xiaoyan. Great choices. So I'll tell you about some of the ones I'm most excited about. And of course, it has to start with the Healy platform trial. I won't spend a lot of time on this because I know you've talked about it in your last webinar, but this really is a unique trial for our field. I would say it's a transformational trial. And the reason is the way that it's set up. So it's also sometimes referred to as a perpetual trial. And the idea here is that you build one protocol and you get all the approvals, FDA approvals, IRB approvals, all the institutional contracts, all of which sometimes takes over a year to get established. And once you build it, now you use that exact same trial to test multiple different regimens. And there can be an infinite number of regimens tested one at a time until the day comes when we find a cure for ALS. And the best sort of analogy that I've heard of, this is from Dr. Sabrina Paganoni, is imagine if you built a stadium like Fenway Park and you played one baseball game in the stadium, and then you never played another baseball game until you broke it all down and built it up again. That's kind of how we've been doing clinical trials in the ALS space for the past 100 years. And so this idea, which comes from oncology, says you build a stadium one time and you just keep playing games in there forever. So I love that idea. And I mean, we have in 4 years since this trial started, completed 4 different regimens. It's about an average of about a year per regimen. There's 2 regimens underway. So to me, it's already been shown to be much faster, much more efficient than the way that we typically do trials. This thing requires an unbelievable amount of administration, and you can kind of see all the different parts of making this trial work on the right-hand side of this slide. There's a lot of different committees. Each new regimen has its own steering and operations committee. But really, again, what I said before, my favorite part is that this group recognizes the importance of patient partnerships. And one of the most important committees on this Healy platform trial is the Patient Advisory Committee. And so again, things I like about this trial, definitely a faster startup for each new regimen. That means we get answers quicker. There's a smaller percentage of patients in this trial on placebo. So there's a 3-to-1 ratio of drug-treated to placebo-treated patients. Now, how do we get away with that? Because we can pool the placebos across regimens. You know, since the inclusion criteria are quite similar from regimen to regimen, you can pool all those placebo groups. Traditionally, you would do more like a 50-50 drug to placebo. Every one of these regimens has to have an open label extension. And again, I love the fact that patients are partners through every step of this trial. The next one I'm going to talk about is one that I hardly ever hear about, and it's surprising because it's one of the few phase 3 trials. for something to make it all the way to phase 3 in ALS, there's got to be a lot of good preclinical and even a reasonably good phase 2 results. Otherwise, a company is not going to spend the enormous amount of money that it takes to do a phase 3 trial. So the drug ibutylast has tons of cool mechanisms of action by which it could affect a lot of different ALS relevant pathways. So you can see here that it can drop oxidative stress markers. It can reduce neuronal apoptosis, which is programmed cell death. It can partially block glutamate-induced neurotoxicity. It can affect the ubiquitin proteasome system. It can alter protein aggregation. It can promote autophagy. It can boost mitochondrial function. It can boost neurotrophic factors, and it can reduce multiple markers of neuroinflammation. And there's a lot of preclinical evidence that this drug works to protect neurons. And so you see, there's a lot of different cell models for different diseases, a lot of different animal models for different diseases where this drug seemed to have beneficial effects. But I mentioned there had to be something exciting about the phase 2 data. And to me, this is incredibly exciting. So the phase 2 study was pretty small, only 51 patients, 17 on placebo, 34 wound up on ibutylast, which in this case was called MN166. So these patients were in a double-blind phase of this phase 2 trial for 6 months. Then they went into an open label extension for another 6 months. Now, when they looked at the progression of the disease as measured by the ALS functional rating scale, including all the patients who got treated with the drug and all the patients who got treated with placebo, they didn't see a big difference. And again, what I said before, it's hard to believe that the way that we're currently doing trials, lumping everyone together, we're ever going to see a home run. We've got to figure out how to subdivide people based on the mechanism of their ALS, or we got to do something called a responder analysis. And that's what jumps out at me about this trial. So this is not the first group who's tried to do a responder analysis. There have been other groups, but I really like the definition that this group used. To me, a responder should be someone who experiences something that is clinically meaningful and also unusual in the natural history of the disease. And so here's one of the definitions they used. A responder or an improver is someone whose ALS FRSR score was higher at the end of a year than it was when they enrolled in the study. That's obviously important. That's what most of my patients tell me they want, something that will give them back lost function. And I can tell you that that's quite unusual in the natural history of ALS. And so if we look at all the patients in the trial, there were more of these responder/improvers on the drug versus the placebo. But it's even more striking when you focus on people who were in the first 5 years of their ALS that came into this trial. In that case, 25% of people who were treated with the drug met that definition of a responder. Nobody in the placebo group met that definition. And even if you allowed those early ALS patients to use NIV, there was still a striking difference. 23% met that definition of a responder or improver, 0 in the placebo group. So again, that really jumped out at me as something I wanted to see more of. And so that's part of why Duke is participating in the phase 3 trial of ibutylast, which is called Combat ALS. Now, there's quite a few rules, as there often is for a phase 3 trial, about who can participate. Some of these are based on what they saw in the phase 2. I mentioned that it was a more obvious effect in people who were early in the course of their ALS. And so they're trying to really hone in on that subgroup. They do limit some of the things that you can take. You do have to be able to swallow the capsules, at least at the start of the study. And then there's some other things that you can't take, some other conditions that you can't have. But this schematic here on the right side of the slide shows you what's expected of patients who decide to participate in the study. There's a screening phase. They go into a double-blind phase where 50% get the drug, 50% get placebo, and that's for a year. And then after that, they roll into an open label extension where everybody gets the drug for another 6 months. So I did want to put this slide in, and I just got permission to be able to share this today. So it's rare that we talk about trials that are underway. And part of that is because we really don't know who's on drug and who's on placebo. Most of the trials that we're doing are double blind. The other thing is it's often hard to tell, even if you have an inkling of what someone got because of a certain side effect profile, it's often really hard to tell that they're benefiting from it. But I'm very convinced that some of my patients, not all of them, but some of them are benefiting from this drug. And I wanted to show you this one patient's ALS FRSR scores. So this is all the way back to when I first saw him, which was 10 months into his condition. We've had scores. And so he enrolled in this trial 14 months into his ALS symptoms. And you could see here for the first, say, 4 data points, there's a pretty clear and pretty significant progression of his ALS FRSR score. So here, month 14 is when he enrolls in the Combat ALS trial. And he's in that trial for 18 months. Look at these scores. There's virtually no change. Then the trial's over, you take the drug away, and he starts progressing rapidly again for the next 10 months. So I was so convinced by what happened to this patient that I actually reached out to the company and asked if they would give me a single patient expanded access program to get him back on it, which they did. So if this trial is of interest to you, you can find the 17 locations that are participating in this around the United States and Canada on clinicaltrials.gov. There's also a main number and a main central email where they can try to help answer questions about whether you're a candidate and what your nearest site might be. So again, just to summarize, I like the drug ibutalas because it has broad mechanisms of action. I think it's possible it could help with multiple subsets of ALS. I love the responder analysis they did in the phase 2 trial. That's meaningful to me. I love the fact that this trial has an open label extension, and I'm convinced from my own experience that it's helping at least some of my patients. I can't help but mention my own study. If I'm not excited about my own study, what am I doing it for? So I have a group of studies that are under the heading Replication of ALS Reversal, or ROAR trials. And these are small pilot trials of these alternative and off-label treatments that are associated with cases where someone recovered from ALS, what I call dramatic ALS reversals. Because most of these products are sold as nutritional supplements, meaning they're generally regarded as safe, you can buy them in a grocery store or on Amazon. And because I'm looking for such a huge signal in these trials, recovery from ALS, not just a slight slowing, I can do some fun things with the inclusion criteria. They can be very wide. I don't need placebos. I can use historical controls. I don't need a lot of in-person visits. And because I know there's a lot of excitement about these types of trials, I'm just publishing the protocols on the website, alsreversals.org. And the reason for that is I know there's people around the world who want to try stuff. They're going to try stuff. So why not put a roadmap out there of what we're doing that they can take to their own doctor and say, you know, could we follow along with this? I've done a few of these trials already. I did one of a nutritional supplement called Lunasin. I did one of a formulation of curcumin called theracurmin. Unfortunately, I did not stop or reverse ALS in either one of those, but I will say I was impressed with what this type of design can do. These were 2 of the fastest enrolling, fastest completing studies in the history of ALS. And they also were done for about $300,000, which is a fraction of what a traditional phase 2A costs. And so I'll get back to something that I said in the beginning. I'm starting to think ALS is more complicated, that it isn't going to work to just lump everyone together and try the same drug in them. I think the biology of this disease probably varies in different individuals. It's just that we don't really know yet how to subdivide most patients. But there's a group out there that might have figured out a way to do it. And so this new trial is going to employ that technology, or it is employing that technology. This is called the ROAR DIGAP trial. So very similar features to Lunasin and theracurmin, except not everyone's getting the same thing here. We're going to attempt to personalize treatment using a technology from a group in Australia called Genius. Their technology is called Deep Integrated Genomics Analysis, or DIGAP. Basically, what it does is it looks at whole genome sequencing It uses machine learning and artificial intelligence to look at the entire weight of the whole genome sequencing. So all the copy number variants, the single nucleotide polymorphisms, the mutations, the variants of unknown significance. And it uses that to determine which pathway is most likely killing that person's motor neurons. And we believe there's going to be 4 categories: neuroinflammation, oxidative stress, disrupted intracellular transport and autophagy, and mitochondrial dysfunction. And so the first part of this trial is categorizing people. Once we have their category, we match them to an alternative therapy that's associated with an ALS reversal that we think hits that pathway. So this is kind of what the schematic of the trial looks like. At month 0, people get consented, they get screened, they get enrolled, they stay on their standard of care, their blood gets drawn. We look at some baseline biomarkers like neurofilament light chain. We draw their blood for the dye gap analysis. Believe it or not, it takes about 3 months to get that back. So they come back at month 3. We draw another set of baseline biomarkers. We give them their dye gap category. We give them one of these 4 supplements matched to their category, and we follow them for another 6 months. And again, we're following ALS-FRSR scores, but we're also following neurofilament light chain and mechanistic biomarkers. So we have a panel of biomarkers for each of those 4 pathways. So this trial is officially the fastest enrolling trial in the history of ALS. We have one last patient to enroll, and that patient is scheduled for early next week. So it looks like the enrollment rate is going to be about 12 patients per site per month. Just to give you some frame of reference, the enrollment rate for most ALS trials throughout history has been 1 to 2 patients per site per month. So I love the broad inclusion criteria, the patient-centric design with few in-person visits and no placebo. I love the attempt to categorize patients and personalized treatments, and I love how fast these trials enroll. So I know we were tasked with talking about current trials, but I can't help but mention a couple things that I see coming, probably in the next 1 to 2 years, because they're so exciting. The first of these is the idea of getting new targets from what I call outliers. So I'm fascinated by outliers. I've always considered myself to be one. But I mean, in 21 years, I've seen 4,000 people come through my ALS clinic. And obviously their progression rates are wildly different. And there's some people that have ALS for 10 years and can still do marathons. Stephen Hawking had it for 50 years. There's a handful of people who have it, progress, and then recover. There's got to be something different about these outliers. And if we understood what that was, we might be able to make everyone have a better prognosis. And so I've got a few things that I found in the past few years with my colleagues at Duke. One of them has to do with the gut microbiome. All of us have literally trillions of organisms in our gut, from our mouths to our stomachs, through our intestines. And it's been known for decades that alterations in those organisms can cause diseases of the gut. And in recent years, it's becoming more clear that alterations can probably influence brain diseases through something called a gut-brain axis. And so we're the first ones now at Duke to show that the composition of the gut microbiome is different in fast versus slow progressing patients from our clinic. What's even more interesting is we can take the stool from those patients and insert it into the colon of the SOD1 mouse model of ALS, and we can change its progression rate. So this is not just an association. It seems that whatever the composition difference is can really affect progression. And so we now have 2 patient and family funded trials that we're building that we think will open in early 2025. One will be of a probiotic. The other will be of fecal transplants. And those are collaborations between the patients that funded these, a lab at Duke called the Valdivia Lab, and in the probiotic case, a company called Bloom, and in the fecal transplant case, the Duke Fecal Transplant Center. Another target that you probably heard about is one that I just published on maybe 2 weeks ago. If I had to say tonight, what's the single most exciting thing that I've ever discovered in my life, it's this. So as you know, I've been studying these so-called ALS reversals and trying to figure out what's different about them. And one thing that looks like it's different is their genetics. So a significant percentage of them have an alteration in a gene that controls the expression of a protein called insulin-like growth factor binding protein 7. It looks to us like the mutation that these patients have results in a lot less IGF-BP7 than most of us have. So this is a very rare mutation in people that don't recover from ALS. And we have a follow-up study underway now with ALS TDI. If I'm right about how some of these patients recovered from ALS, we should see a signal in the ALS TDI ARC. There should be a relationship between how much of this protein patients have and how fast they progress. The less of it you have, the better you should do. I should have the answer any day, but if that turns out to be true, then this is going to be my most exciting trial that I've ever designed, which will be one to try to knock down IGF-BP7 to see if I can stop or reverse ALS. I love this trial from my colleagues at the UK. It's called Experts ALS. So Chris McDermott and Martin Turner, these are guys I've looked up to my whole career. And they've got something that's kind of like a platform trial, except it's not using clinical outcomes. So what they're doing is putting small groups of patients on different drugs, and they're looking to see if neurofilament light chain levels drop. And the nice thing about that is they might be able to tell within just a few months if they're on the right track with the drug. So instead of 6 or 12 months, they might be able to do a trial in 2 or 3 months. So this is how it could work. So this graph on the left-hand side of this slide is what happened in the pivotal tofersen trial to neurofilament levels. And so you see a curve that's blue, you see a dotted line, and then you see a curve that's red. The blue curve represents patients who got randomized to placebo. The red curve is patients randomized to receive tofersen. The dotted line is when they moved from the blinded phase to the open label extension. In other words, after that dotted line, everybody got tofersen. And I think what you can see here is the patients in the blue, their neurofilament light chain level didn't really change for 28 weeks. Patients on tofersen, it plummeted down to about half of where it started in just 12 weeks. And even if you looked at the patients who got placebo from the beginning, once they started getting tofersen, their neurofilament light chain level plummeted as well. So I mentioned earlier, this is incredibly important because in the case of tofersen, at least, it predicted a much later profound clinical benefit. And so I think that these guys are really onto something. I think that they're going to be able to find drugs much faster that are promising for clinical trials. And I see tremendous potential in this idea. Specifically, I see 2 things that I've been dreaming about. One is the first truly decentralized ALS trial. I mean, there's no reason why we couldn't do a trial like this for people all over the world in their homes, where we send someone out to draw their blood. We wouldn't need to have them come in and measure anything fancy. We could just measure this. Even better, this could allow us to do something which is called high throughput drug screening in people living with ALS. So high throughput drug screening is something that industry has been using for decades. Basically, you develop a target that you think is relevant for a disease, and then you dump thousands, tens of thousands of drugs onto that target to see which ones hit it. Well, guess what? There already are literally tens of thousands of N of 1 experiments going on all over the world in people with ALS right now. Like almost every one of the 500 patients that Dr. Li and I follow in the Duke ALS clinic, they're on something. Most of the time, it's either one alternative or off-label therapy or a combination, sometimes of up to 30 or 40 of them that the patient has decided upon. Wouldn't it be awesome if we had a way to easily measure which of these things might be most promising by checking neurofilament light chain levels in everyone at regular intervals? So I hope that's where we're going to go with this. And then the last initiative I just wanted to throw out there, it's got a question mark because I don't know if it's going to happen. I hope so. It's something that I would call the lighthouse. And I mentioned in the beginning that I think this field has a critical need for widely available, reliable, personalized education, especially about research and experimental options. So what I'm proposing here is teleeducation. It's not telemedicine. So in this program, people living with ALS would be able to schedule a free 30-minute video chat with an expert. And they could ask that expert questions about anything they wanted, including, could you help me find a trial or an expanded access program? Could you match me up to a promising supplement? Because I know that not every clinic is doing this for their patients. And, they could get a written summary from the expert with useful links afterward. And I really feel like a tool like this would empower people with ALS with new options that they could take to their clinic, talk to their own doctor about. And most importantly, I think it would give people a lot more hope. And all I'm looking for now is a patient advocacy group partner who wants to host it. I've pitched it to a bunch so far, and so far no takers, but keep me posted if it's something that IMALS might be interested in. So just to kind of summarize, trials, they're the only way that we're ever going to find better treatments and hopefully someday find cures. They're not easy, but powerful collaborations between patient advocates, patient participants, and researchers are moving us forward at a faster than ever pace. And I could not be more excited about where things are going for us in the next few years. And I'll just conclude with some thanks, most importantly to people living with ALS. Your advocacy is why all this progress is happening. Your participation is everything. I mean, we can design the smartest trial in the world, but unless you sign up and help us with it, we can't go anywhere. The scientists who are out there coming up with all this brilliant preclinical work, the clinicians like Dr. Li, still in her office, it's almost 8 o'clock Eastern time. Thank you so much for your sacrifice. And of course, all the sponsors. So I'll stop here and we'll take some questions.
All right. First question is, Diane, when someone finds more than one clinical trial that they might qualify for, they often want to know which one would be better for them. What advice would you give them?
Yeah, Michael, it's unless the trial is targeting some specific gene or biomarker that we know is abnormal in that patient, there really is no way to know which one's better. If we knew which one was better, we wouldn't do the other trial. We would focus all of our attention on the one that we thought was better. So what it comes down to is which one's more feasible. If one trial is 15 minutes from your house and the other one's 500 miles from your house, I would do the one that's 15 miles away. A bird in the hand is worth two in the bush. Do the trial that's going to create the least chaos for your life. Do the trial that you think you can finish rather than the one that you might only make 1 or 2 visits to.
Thank you. Next question is from Nancy. Any preliminary info from the 42 Italian patient? Sorry, any preliminary information from the 42 patient Italian trial on FMT?
None that I've seen. It's kind of a mystery what's going on with that trial. I haven't seen any updates in a long time. And that's part of why we just decided to go ahead and launch our own trial. I'm not even entirely sure what the protocol for that trial is. Like, what I've learned from the Duke Fecal Transplant Center is it's much more complicated than I thought. It's kind of like stem cells. Like, I know I've had some patients who went somewhere and paid a lot of money and got 1 injection of stem cells. That's never going to work. I mean, stem cells, if they're ever going to work, they're going to have to be given over and over again. And there's a lot of questions that are going to have to be answered. But one fecal transplant is not going to change your gut microbiome. It's very clear that we're going to need to do something over and over again. And so I'm learning from them about different ways to change the gut microbiome. But I have patients even now asking me, can you get me a fecal transplant? Well, 1 fecal transplant, it's not going to do anything for you. It's not going to even change your gut microbiome. We would have to wipe out your existing microbiome, probably with something like an antibiotic purge, as well as a colonoscopy prep. And then we would have to probably do, I'm guessing, monthly fecal transplants for at least 4 to 6 months to change that gut microbiome. So it isn't going to be easy, but we're going to try it.
Next question is, of the 40 genes involved in ALS, how many are on the same chromosome?
You want to take that one, Cheyenne?
Yes. Actually, I'm not aware of any two of those 40 different genes or one same chromosome. But also, I'm not sure about that, how to answer that. So I didn't really look into it, but that's a really good question.
We'll do a little
Maybe I
research and get
can,
back to you. We'll do a little research on that one and
yeah,
get back.
maybe I can do a little bit of research and get back to you later, but that's a really good question.
Sounds good. Next one is, why can't the results be reported in a more timely manner once the clinical trial is over? I find the lack of urgency troubling.
Let me tell you, you're not the only one who finds it troubling. That phrase, lack of urgency, is, if I'm ever beaten and I ever decide to leave this disease before we find the cure, it's going to be because of that. And the reasons that you get are so incredibly frustrating. I mean, it takes forever for anyone to hear the results. And then, once the results get out there, if you want to publish it in a good journal, many of the journals will embargo. What that means is you're forbidden to talk about it. So, we published our IGF-BP7 finding in the journal Neurology. It's a great journal. It's really hard to get articles in there. Most of the stuff I send there gets rejected. So I was really proud to get it in there. The downside is, for 6 months, I was not supposed to talk about it. And it really bothers me. I don't actually see a good reason for that, other than you're trying to sell downloads. And the thing about it is, most of us pay the fee to make our article free open access anyway. So even that argument sort of fails to hold water. And I mean, go back and look at the AMX 0035 data, how long it was before that trial was done and the paper came out. I think it was about a year. And again, I think that one was published in New England Journal, which I'm sure also has a huge embargo policy. So yeah, I mean, it's the lack of urgency does not end there, I'm afraid. Part of the reason why it takes so long to get anything open is because there's a lack of urgency in almost every part of the trial opening process. I will say the FDA is actually not the problem. They're pretty fast about approving things. But like when you have lawyers at institutions where one sends a version of a contract to the other, and it's 3 weeks before a response is sent back to the first lawyer to say, what can you live with? And then it's another 3 weeks before that person gets the contract back. It's outrageous. And I'm sure that they're understaffed and they're overworked, but I feel like many people in this field don't have the perspective on this disease that they need. Like, a piece of paper is actually someone's function. It's actually someone's life. It's their hope. And I've talked to some of the lawyers at Duke about that. I said, if this was your spouse waiting for this drug, hopeful for this drug, the only thing that they might be able to get their hands on that might slow, stop, or reverse their progression, and you held on to that piece of paper for 3 weeks, how would you feel? So I don't know what to do about it. I'm doing my best to scold people and call them out, but I mean, thankfully we have things like the Healy platform trial that help with some of this lack of urgency by setting up a single trial. But I don't know how to bring all these people, to the table and get them to understand what this disease is. I don't know how to bring journals to the table to get them to understand that science requires us to be able to talk about our results, and ideally in a timely manner, not waiting 6 months for an article to come out before we can start, collaborating and talking about the research. So I don't have an answer. It's frustrating.
Next question was partially answered by Diane. Well, it was answered by Diane, but I'll still read it just in case you have a different answer. What website resource do you typically find clinical trials? I am a caretaker of a recent ALS diagnosis. Diane responded with, try IMALS's Signal and ALS TDI's Trial Navigator. Both can help you find trials.
Yeah, those are great. I mean, clinicaltrials.gov is another one. It's comprehensive. It's difficult to use. I mean, I will go back to what I said earlier, and that is, in my experience, many people that come to my clinic are not the most internet savvy. You know, like I think about my parents. If I sent them home from clinic and said, well, you know, there's these websites, go look up, see if you find any trials that look interesting to you and let me know what they are and we can talk about them, they would be lost. They wouldn't know how to do that. So again, I think this service that you could have where somebody could dial into an expert and say, I want to do a trial. Can you help me find my options? Here's where I live. Here's how long I've had ALS. You know, here's my last pulmonary function test. They could actually go through those websites for you while you're on the phone and try to find 2 or 3 things that you could think about and talk to your own doctor about.
And our last question, I am probably going to butcher the pronunciation, but you'll all forgive me, I'm sure. Any thoughts about conservative iron? Okay, Chelathion, I don't know how to say it. Chelathion with Deferbone, Deferipone. I understand being studied in France.
Yep. You want to take that one, Cheyenne, or you want me to?
I'll defer this to you.
Okay. Yeah, I mean, it's an interesting idea. I have to say, whenever someone asks me about a new therapy, I start with, what's the evidence that the target is really involved in ALS progression? And I'm not convinced that any metal is driving the progression of ALS. Not right now. Maybe somebody will change my mind, but I've also seen a fair number of those 4,000 patients that I take care of have a variety of different types of chelation. I've never seen any obvious benefit from any of them. Now, this is a new way to do it. So it's possible that this is going to have an unexpected effect, but it wouldn't be on my list of most promising things right now that I'm looking forward to seeing the results of.
Great. Well, we've gone beyond our 1 hour. Thank you, Dr. Bedlack and Dr. Li, for hanging in there and answering the questions that people had. Michael put a comment that he will send the links that were shared in the chat to people. Laura just put a thank you in the chat. Oh, we have one last raised hand from Dan. So getting in under the wire. Go for it, Dan.
I'll make it really quick. I just want to say to both of you that every time I have the opportunity to listen to you, I'm struck by the clarity of what you have to say, your enthusiasm for the science, and just as much your compassion for those living with ALS. And for that, thank you, thank you, thank you.
Well, that means a lot, Dan. Thank you. And like I said, I feel very blessed to be in this field. I'm surrounded by passionate, smart, creative people. There are definitely some frustrations like we talked about earlier, but you know, when I lay in bed at night and I think, you know, of all the good things in my life, they always far outweigh the bad. So I'm happy to be here, and I think Dr. Li feels the same.
Yeah, thank you for your nice words. It's a pleasure to be here today.
Yes, lots of nice words in the chat, and lots of thanks and gratitude for you. So again, thank you, and thanks everyone who took the time today to join this call and to listen to Dr. Bedlack and Dr. Li. And thank you for those who've participated or are participating in clinical trials. Couldn't be done without you, and recognize the commitment and sacrifice that it takes. So thank you very much. And I think we are probably good to call it a night. Thanks, everyone.
Bye, everyone.
Bye-bye.